All right, our next presenter is Professor Dr. Adrian Covaci. Dr. Covaci is currently a professor of environmental toxicology and chemistry at the University of Antwerp, Belgium. He is currently the director of the Toxicological Center in the Department of Pharmaceutical Sciences. After acquiring his PhD in chemistry in 2002, he went on to become a full professor in 2012. Dr. Covaci's current fields of interest are the application of advanced mass spectrometric techniques to the human exposome, human biomonitoring of emerging contaminants, and metabolomics and human metabolism of xenobiotics in vitro and in vivo systems. He has co-authored more than 850 articles in peer-reviewed journals and has an H-index of 105. His papers have been cited more than 42,000 times, which has earned him the title of highly cited research for 9 consecutive years between 2015 and 2023. Since 2011, Dr. Covaci has been the Belgian representative in the Division of Chemistry in the Environment of the European Chemical Society and a member of the Belgian Health Council since 2022. He is co-editor-in-chief for Environment International, and between 2012 and 2022 was an associate editor for Science of the Total Environment. Welcome, Dr. Covaci. You may start your presentation now.
All right. Thank you very much, Ben, for the kind introduction. And thank you for having me here. It's a great pleasure to give a talk. I have chosen a topic which is a little bit more focused this time, but nevertheless very novel and maybe on the technical side, as I'm going to explain how the ion mobility helped us really identifying new compounds. So over which compounds are we talking about? So these are the quaternary ammonium compounds, so the QACs. And they are used as active ingredients in more than 200 commercial products. I think that I took this information from EU and EU source, so probably there might be other products in the US or Asian market, and they are mostly used as disinfectants. And there are currently individual QACs, more than 800 currently in use. And they are cationic surfactants. So you have here the structure of different groups, so 3 main classes, and they all have something in common, and they have this quaternary ammonium site, which has different substitutions. So the first group has 3 methyl substituents and the long chain, and the BACs have a phenyl, actually a benzyl ring, and 2 methyl and the long chain, and the last group, the DDACs, have 2 long chains and 2 methyl on the nitrogen, on the quaternary nitrogen. And so these cationic surfactants are used in cleaning products, as you can see here, biocides, pesticides, food industry, medical care, pharmaceutical products. So really a lot of applications. So actually, we might expect that the human exposure to those compounds is rather important. And I have selected, there is not so much about the toxicology, I have to be honest. So I have selected a few things which I think they are relevant in the context of the human exposure, and this is skin irritation, asthma-related symptoms in occupational settings, so people using a disinfectant like cleaners, for instance, and also increase in inflammation, cytokines, and decrease in the mitochondrial function. So now the study setup, we have selected 2 types of matrices to work on. So one relates to our external exposure, so the indoor dust, and the other one relates to the internal exposure. I will explain later when we get there. So for the indoor dust, this is a study that we have conducted around 2020, 2021, rather a limited number of indoor tasks selected in Flanders, so the northern part of Belgium, and we had a quite nice distribution between private residents and also different other types of public buildings. In the presentation, I'm not going to discuss the differences in the compounds between those different types, but this is also part of the paper that you will see later on appearing. So just very briefly, the sample preparation was rather simple. So we have used 20 milligrams of dust extracted with methanol, evaporated methanol, and then reconstituted and then analyzed it. And of course, in referring to the previous question with Georgie's, of course, we add also labeled internal standard, as many as possible as it is available on the market. And so we add this label internal standard in order to quantify the native compounds. So that was somehow the answer to the question. So to quantify the native compounds present in the sample, and of course using calibration curves based on the native and the internal standards. And so we initially started by looking to these three classes, and we had around 21 compounds, if I'm not mistaken. So 21 compounds, so 8 from a first class and 7 from the BACs and 6 from the DDACs. So this is what we could find as native standards. These samples were quite precious, so we also have done a lot of other things. So we looked for many other compounds, which I'm not going to discuss in this presentation, but there is a lot of information that we have subtracted from this sample. So initially we thought, okay, let's analyze using a quite sophisticated technique. So we wanted to analyze these dust extracts by liquid chromatography coupled with high resolution MS. So in this case, it's a QTOF, which offered the possibility of having MS spectra. So as identifiers, so you have the retention time, the first dimension, a mass spectrum, a second dimension, and then an MS spectrum or a fragmentation spectrum as a third dimension. And this is a quite current procedure in the literature. And since 2014, we and many other people are using this annotation scheme or identification confidence scheme proposed by Szymanski. So 2014, I said that, in which there are different levels of identification possible, and very important, level 1 is confirmed structure with a reference standard. So having already 21 compounds as a reference standard, we could confirm the structure of several of these compounds. Anything for which we don't have a reference standard will be at lower levels, actually level 2, level 3, in which we have an possible annotated structure using the mass spectra or the fragmentation spectra. Now, because our system offers the possibility of having the ion mobility, we have introduced this ion mobility dimension somewhere between the chromatographic separation and the MS or MS analysis. And in terms of time, as you can see here, so the chromatographic separation is in seconds, the ion mobility is in milliseconds, and the mass spectrometric analysis is in nanoseconds, so it also fits very well. And so this ion mobility is a gas phase separation technique of ions, which are already formed after the molecules have passed the ion source. So the molecules are ionized in the ion source, and they enter the drift tube, so the ion mobility part of the mass spectrometer. And so this drift tube, as you can see it here, so the ions will move from left to right, and there is a current of gas, of nitrogen gas, moving in the other direction, which is somehow slowing down the ions. And the time that the ions are spending in the drift tube is called the drift time, and it's determined by the size, shape, and of course the charge of the ions. And this type of separation gives us the possibility of separating isobar ions or also isomers. So it's a very good and very fine separation technique. There are different types of ion mobility. I'm not going into details. So besides the drift tube, there is also the traveling wave ion mobility, and also the trapped ion mobility by other vendors. And what is very interesting is that I'm going to put it here. So the ion mobility and especially the drift time, so the time that an ion spends traveling through the drift tube, this drift time is directly proportional with this CCS value. So it's a collision cross-section value, which is expressed in square angstrom, which is the projection on a two-dimensional plane of all possible molecule conformations. So that means that this parameter is a property or another identifier of a specific ion, and like this we can also use it in the puzzle, which will help us in the end to annotate or to better annotate a molecule. So now, the scheme of Szymanski 2014 had to be a little bit adapted if we want to include these CCS values in the annotation and in the identification, actually, of the different compounds. And here you see the novel scheme, which was published in 2020 by Selma et al., which includes this CCS value. I'm not going into detail, otherwise it will take me another 20 minutes to explain only that particular scheme. But the point is that using an additional identifier like the collision cross-section value helps us to better annotate or identify a chemical. And then, of course, there are also databases which are starting to be more and more populated with compounds which have CCS values measured experimentally, most of them based on reference standards available. But at the time that we started our work, there were no CCS values available for the QAC. So we thought, okay, all right, let's maybe make some experiments and have already some reference CCS value for at least for the compounds that we have as standards. So in general, for this work, what we wanted to do is to do a kind of target screening for the list of the 21 QACs that we had, and then to see which of those ones are present in the dust, and eventually to do some quantification. We also created a long list of a suspect list of known QAC. I think we had almost 300 QACs possibly present in the dust samples. And we have tried to quantify / semi-quantify some of the compounds that we have identified. And of course, we were very interested in go deeper into this ion mobility technique and also acquire as many CCS values for all the QACs that we can annotate. And then part of our work was also to calculate an estimated daily intake of the compounds that we could quantify. And this estimated daily intake, for the people who do not know, is based on the concentration measured in dust multiplied by the ingestion amount. How much dust do we eat for most of the time? So it's mostly oral ingestion. What's the fraction which is absorbed in the body, and divided by the body weight. And we use this estimated daily intake to calculate hazard quotients. So we divide the estimated daily intake by reference doses if they are available for the compounds of interest, and the hazard quotient above 1 would indicate a potential exposure risk for the target population. Now let's go to some of the details. So in first phase, we looked for the compounds. We look after the compounds for which we had the standards of the 21 compounds, and these compounds have been found with various detection frequency, but most of them were at 100% detection frequency in the dust samples. And then we calculated the concentration of these compounds based on different calibration curves. And you can see here the distribution. And what I want to point out is that for all compounds, excuse me, so for all compounds, we have calculated an hazard coefficient below 1 in this dust sample. So it means that the presence of these compounds at individual level, so the interpretation is at individual level, will lead to no, there is no potential health effect at all. However, there is very little known about the mixture effects for these compounds. So this is something that probably we will look into the future. And maybe one more thing that I should tell here, that we have identified compounds, or could measure compounds from all the 3 classes that we started at the beginning. Now, how do we fit the work on the ion mobility into this? So for the compounds for which we had the standards, we have acquired the CCS values. So this is on the y-axis, the values, as I mentioned, in square angstrom, and we plotted them against the mass to charge ratio of the ions. And all the gray points that you see here are different other classes of compounds that we have analyzed. I think that we had around 150 compounds that we have built this database. Now, if we zoom in a little bit in the QAC region, we see different trend lines, we would call them. And these trend lines are composed of the compounds that we have the standard. So for instance, if we take the DDACs, we see those orange or dark orange points. And these are the compounds for which we had the standard. So with the long chains, the symmetric long chains, so C14, C14, C16, C16, and C18, C18. Now, in the same plot, we had those compounds with light orange, and maybe I should have started telling that for the BAC the same. So for compounds that we had the standard, so the green ones, we could identify them quite easily. So the length of the chain, but we had also some values, so the blue ones for which we didn't have standard, and we thought that there might belong to this class, so the blue points might belong to this class of BACs, and the light yellow orange would belong to the class of the DDACs because of the apartness on the trend line. And so based on this property, and of course, based on the MS spectra and the masses, so all the other identified, and the retention time, and so the MS and the MS spectra, we could identify several compounds, so like the C18 chain, the C13 chain, and the C20 chain, for which we didn't have standards. So for these compounds, the annotation is at level 2. Meanwhile, for the green compounds, the green dots, the identification is at level 1. So this is for the BACs. We have identified at least 3 new compounds. And then for the DDACs, as you can see clearly here, we have identified several other compounds, including one mixed compound. I will talk about that later. One mixed compound that we could not really separate it chromatographically on the LC. What do we still see here? Compounds which are orphan. They are not belonging to any class, as like this benzethonium present here. And as I mentioned, the collision cross-section value as an additional identification parameter. And we have identified also a new class of compounds, so the dimethyl ethyl alkyl ammonium, these DHCs with lower masses and also lower CCS values. And so this annotation is again at level 2 or 3, depending on on the compounds. So of course, for these compounds, we didn't have any reference standards to confirm them. Now, I was talking about that mixed compound. So for that particular compound, so it was one larger peak on the LC for which we had a very strange fragmentation pattern. So we had different fragments belonging to the longer and longer chain, as you can see here. That's C12 chain, 14, 16, and 18 chain, starting from the same parent ion. And then when we apply the ion mobility, so their drift time, or the drift time of these two compounds through the ion mobility section, we clearly see two different peaks. And so we could confirm that this is a mixture of compounds having the same parent ion, so the same mass. Now, so this is a little bit of recap what we have done. So we had this targeted QAC, so the initial 21 compounds that we had available, and then we have identified through this suspect screening workflow, we have identified this new class of DEACs, and then also new DDAC compounds, and then new BAC compounds. So these are all newly identified, or actually annotated, because they are still at level 2 of identification. And so, very interestingly, some of those compounds, so I picked up this with uneven, even chains, but not symmetrical chains, so the C16, C18 DDAC, and with the detection frequency at 100%, and actually also a concentration which was pretty similar to the compounds for which we had the standard. So this is not a minor compound, this is equally important as the compounds for which we had the standards. And of course, we have recalculated the hazard quotient again for individual compounds, and that was still below 1. Now, in the second part, and I still have maybe 5 minutes, if you allow me. So in the second part, we wanted to understand what happens with those compounds once they enter the body. So for many other compounds, we would know to what they are biotransformed, but we had no clue what happens with these compounds. So we have tried to do this artificial metabolization procedure using in vitro transformation with human liver microsomes and also with S9 fraction. And in this case, we will form metabolites from phase 1 and phase 2. And so we have selected 3 compounds from these 3 different classes for which we had the reference standard. Of course, this is very important. So let's start with the first one. So with the 1010 DDAC, and we have observed 13 metabolites, or possible metabolites coming from different metabolism reactions. So you have here which type of metabolites we have observed, and we believe that they are looking in that way. So when monohydroxy, of course, we have no idea where the hydroxy sits on the chain, or dihydroxy, and then a hydroxy followed by elimination of 2 hydrogens. So it can be a keto, it can be an aldehyde. We also don't know the position at the same, so an keto and an hydroxy, and then again then hydroxy, and then 2 oxygens, so probably an carboxy, and so carboxy and an hydroxy, and then carboxy, so we'll make it more difficult, and carboxy and a keto, and maybe a trihydroxy, and then eventually in dicarboxy compound. So a very complicated metabolism pattern, and in many cases we could identify the class of metabolites, but not the position of the hydroxy or the keto group. And so we have also put these extracts through the ion mobility to understand what really happens. And this is the plot we got. So it's a little bit more schematic, but we have on the y-axis the CCS values, and then the M/Z on the x-axis, and then we could see a clear decrease in the CCS value. So it means that the smaller area or a projection of the compound, and it stays more or less the same. So the idea would be that compounds will start to fold a little bit better and then have compact structure, probably because of the presence of this oxygen and then attraction of one side to the other one. So just more compact structures. We also wanted to go a step further to understand if those compounds found in a synthetic way, so in the human liver with the human liver microsome, so in vitro, if we could find those compounds in vivo, so in a urine sample, that we had healthy volunteers from another study, and would pick up the random 4 urine sample per participant, and this is what we have identified for this specific compound. So starting from the parent 10 DDACs, we have identified a number of 5 possible metabolites with these potential structures. And just I have 2 more slides of data. So one slide looks at the biotransformation product of the 13C12 BAC, as you can see here. And for these compounds, for this compound, we had obviously less metabolites because we have only one chain, and in the same way, so the monohydroxy, a dihydroxy, a ketoaldehyde, or a carboxy. And luckily, some of those compounds we could identify with standards, which were recently bought. I think that 2-3 months ago we bought them from CIL, so the hydroxy metabolite and the carboxy metabolite. And here is an example of the standard. So the upper part, upper chromatogram is the standard, and then the lower part is an HLM, so the liver microsome extract, where we could see that we have probably 2 isomers of the hydroxy BAC. And so this type of identification was possible only because we had the standards, and of course the additional hydroxy metabolite. We didn't have the standard, but we assume that it's an isomer just because of the similar mass spectrum. And now having these native standards and the corresponding internal label standard, we can even quantify the metabolite in the real urine samples. So maybe I'm going to the conclusion. So I hope that I convince you that this class of compound are quite interesting to look into. There are very few studies in humans related to the QACs, and actually they are quite a lot used in different products. I encourage you to look at your home which kind of disinfectant products you have and try to find them on the label. And then of course, to further identify them, we need ion mobility, and of course we need quantitative data for exposure assessment. Using the CCS trend lines might help predict some of the compounds, or help in the identification of some of the compounds. And of course, for the in vitro biotransformation, we have confirmed that oxidation is a main pathway, as for many compounds having such kind of alkyl chains. And then we could partially confirm in urine samples. And up to now, we didn't look to other excretion pathways, but there are indications that some of those QACs may be also excreted in feces, transformed or untransformed. So I'm going to stop here by thanking in particular Lydia. This was part of her PhD, and she has worked very hard in getting the ion mobility responding in the way that we wanted. And then of course I thank also the members of our team, and thank you very much for your attention. And
Thanks so much for sharing your impressive work, Dr. Covaci. I really believe that the study of disinfectant compounds is an area that we should see even higher interest in the coming years. So we really appreciate your presentation about this really interesting topic. I think in the interest of time, we're going to move along on. So I'll invite anybody that may have questions to please once again go ahead and enter them into the Q&A section box. And
I will answer by typing in the Q&A
yes,
box.
exactly. Great. Thank you very much.